Thank you, and welcome to this first talk in our fourth series of expert briefings that PDF, Parkinson's Disease Foundation, has been running now for the last 4 years. The title of this talk is What's New in Genetics and Parkinson's? Always one of the most popular and interesting and challenging of the topics, and it's no surprise that we're welcoming already almost 1,000 people who've pre-registered for this talk, and others will be joining us, so we welcome them all. And I also want to make a couple of announcements of gratitude. First of all, for our sponsors, we have two companies, Abbott Laboratories and Teva Neuroscience, who have been sponsoring this series. We're very grateful to both of them. The responsibility for the series, of course, is entirely ours, but the responsibility for funding has been mostly theirs, and we certainly appreciate this. This series of educational seminars is only part of the work that PDF does. If you're unfamiliar with our work and you'd like to check in with it, please visit our website at www.pdf.org. It's the shortest domain name in the business, and check us out and see if there's anything else you'd like to be in touch with us about. It's my great pleasure now to introduce our guest speaker today, Dr. Dr. Farah. He is the Canada Excellence Research Chair in Neurogenetics and Translational Neuroscience at the University of British Columbia in Canada. He was previously for many years at the Mayo Clinic in Jacksonville in Florida, and is really one of the outstanding genetics specialists in the Parkinson's field in any country, and we're just thrilled to have him here today. He will speak for about 35-40 minutes. You're probably familiar with this, those of you who've been with us before, to slides, and then we will have time for questions afterwards. So please don't be bashful in emailing us your questions, and we have a team of two here who will be quickly going through them and preparing them for me then to edit and combine and direct them to Dr. Farah in the last part of the show today. So welcome. Thank you so much for joining us, Dr. Farah. This is really a great privilege for us. He's an altogether wonderful leader, and the only possible misgiving I have about a total accolade here is he has a rather strange accent which betrays the homeland that we share.
Okay. Thanks very much, Robin, for the introduction. It's a great pleasure to tell you a little bit about what's going on in the genetic world in Parkinson's disease, and there's a lot of ground to cover, so I'll get right to it. First of all, I think it would help the listeners to appreciate why we're doing this and what genetic has in terms of what can it contribute to our understanding of Parkinson's disease. And really, I'm going to preface that with talking about the symptoms and the treatments for Parkinson's disease. As many know, levodopa, the main treatment for Parkinson's disease, works very well, but it doesn't work forever. And it is associated with disabling side effects and problems, and also patients tend to progress in their disease and tend to have to take more levodopa with time and have more fluctuations and perhaps more problems during the course. What we're after is to try and find a better understanding of what's really going wrong in the disorder in the brain, and what are the fundamental causes of that. What we'd like to do is to help, well, two things. One is to predict, and the other is to prevent Parkinson's disease in its progression, perhaps prevent it altogether, in, for instance, individuals who may be more susceptible to the disease. So I'm going to tell you about three things, not necessarily in the order on the slide, but we're going to discuss the latest findings in genetic research of Parkinson's disease more towards the end of the talk. I'll tell you a little bit about what genetics research, or how to interpret genetic findings in Parkinson's disease more towards the beginning of the talk, and throughout the talk I'm going to tell you about the new treatments and the excitement there is in developing new medications around these targets in big pharmaceutical industries. I'll move on to the next slide. Can I control this? Okay. There we are. Okay, so hopefully you should see this slide 4. The analogy here I've drawn is with a car engine, and And really, this is the problem. I think in Parkinson's disease, we don't really understand what the components are. For instance, if you have a problem with your car and it just doesn't start in the morning, or it's slow or sluggish, you've got a pretty good roadmap. You can look under the hood, under the bonnet, and see the different parts of the car engine, whether it's the spark plugs or the carburetor or the battery, and you have a series of diagnostic checks that you can do to see, really, to pinpoint what the problem is. For instance, maybe it's a dead battery, and you can quickly remedy that by charging it up or putting a new battery in the car, and lo and behold, it works again. You've fixed the problem. In Parkinson's disease, we'd like to do the same thing. It's not enough to just alleviate the symptoms, I think. So what I'm going to tell you about is how we're trying to basically find these different components of this, if you like, motor for dopaminergic neurons in particular, and then how these different components can then provide new targets for therapeutics to remedy the problem. So in the top left-hand corner there, I've got a picture of a car engine with all its component parts, and in the bottom right, I've got a cartoon of a neuron. So the bottom part of the neuron here, I don't know if you can see the mouse on the screen, but the main cell body of the neuron basically has lots of small processes or dendrites that stick out of it that receive input from other neurons, other brain cells in the vicinity of that cell body. But it also has an axon, this long filament that basically reaches from, when we're talking about dopaminergic neurons, from the substantia nigra, the black substance in the brain, all the way to the striatum, the region of the brain that controls the initiation of movement. So the axon basically ends in a synaptic terminal here, synaptic bouton. And typically, for a single dopaminergic neuron, they have something in the order of 300,000 of these terminal boutons in the striatum that innervate the striatum. So it's a very this is a very simple cartoon for a very complex cell. I've illustrated on the cell a number of the different genes that have now been implicated in Parkinson's disease. And I'm going to tell you a little bit about the methodology, the way in which we find them, and then I'm going to tell you, but to focus more on what we're doing now in terms of joining the dots. Okay, we find each of these components, be it the battery, be it some spark plugs, be it the alternator. We're trying now to figure out how they connect together to make this engine run. So in terms of the approaches that we use, there are two. It's quite simple, really: linkage and association. And the findings, depending on which method is used, have different ramifications, and I'll illustrate that. One thing I'd like to point out is that everybody with Parkinson's disease in the world can take part in genetic studies, and it would be phenomenal, absolutely amazing if they would. We do these studies here in British Columbia. There are many, many groups around the world doing these types of studies. I wouldn't think it's too much of a problem, maybe for epidemiologists, but not for geneticists to be engaged in with more than one group and perhaps even doing similar work, although you don't want to duplicate too much effort. But it would be wonderful if everybody would get engaged, both patients with Parkinson's disease, family members, as well as their spouses, their unaffected spouses who would act as normal subject controls. So the two methods are linkage versus association. And to give you some illustration, on the next slide, I show you on the left-hand side, these are the genes that have been implicated through linkage. And by that, mutations or single nucleotide changes, so one change out of the 6 billion nucleotides we inherit, maybe an adenosine to a cytosine, or we have 4 nucleotides, A's, G's, C's, and T's. And a change of one of these in one of these genes may be the culprit for Parkinson's disease being inherited down the family line. So the first gene identified for Parkinson's disease was alpha-synuclein, SNCA is the acronym for it, the top one on the slide. The first mutation was a guanine to adenine change at position 209, which led to an amino acid change of alanine to threonine at position 53. You don't have to remember these things. I have to remember these things, but But what I wanted to point out is the precision in understanding that this specific base, one of 6 billion nucleotides, is the culprit in these families, in these affected family members with this particular form of Parkinson's disease. So synuclein point mutations were initially identified in families in Conterzi, Italy, and in Greece. But several other mutations, and I'll get to that, have been discovered since. There are many other genes on this list, as you can see, and each of these, when you have mutations in these genes on the left-hand side, basically are sufficiently deleterious, to put it that way, to basically promote the disease down the family line. So basically, for instance, an alpha-synuclein mutation in a parent gives rise to a 50% likelihood in the children of that parent that they will go on to manifest Parkinson's disease if they live long enough. So basically 50% of each generation may be affected, and males and females equally. Some of these mutations are inherited in a recessive fashion rather than dominant, like PINK1, DJ1, Parkin, whereby 1 in 4 individuals may be affected, and neither parent may seem to be affected. Basically, they both carry mutations, but you need to inherit 2 mutant copies of the gene, one from your mum, one from your dad, to manifest the disease. On the right-hand side are susceptibility loci for Parkinson's disease, and these are found through association, not through linkage. And basically, this is a different technique. So instead of doing a family-based study, this is a study where many people with Parkinson's disease and many control subjects basically contribute DNA samples to very large studies, typically involving tens of thousands of people. And basically, we look for differences in the frequency of variants, common variants that we all have, but we see whether these frequencies are most different between people with Parkinson's disease and people without the condition. This is nominated now something in the order of around 20 different regions of the human genome in Parkinson's disease, some of which include genes on the left-hand side that are deleterious and for which we know that single point mutations can also segregate down the family line. You can get an update on both of these things. Basically, if you look at gene tests, it's an NIH-sponsored site, you can find out more about predictive genetic testing for Parkinson's disease, for mutations that segregate down the family line. Whereas if you look at, for instance, PDGene, on the right-hand side, you can get an up-to-date meta-analysis of all of the variants that have been implicated in Parkinson's disease through candidate gene and genome-wide association studies. I should say that the genes on the left, because they segregate down the family line, the mutations on the left basically have a larger contribution towards disease risk. The genes on the right typically have a rather small contribution to disease risk. They may double the odds of getting the disease, for example. But if the odds of getting the disease are only 3 in 1,000 anyway, so the odds when doubled are 6 in 1,000, it's relatively small increases in risk. But nevertheless, these findings are very interesting to the neuroscience community. I thought I'd better talk a little bit about personalized medicine and how genetics affects it. And the illustration I was going to give, because I think it might come up as a question, is around the idea of a lot of these new companies that are popping up that do direct-to-consumer genetic testing. I think this is a noble effort, really, for the most part. Basically, you can send your DNA to a company like Navigenics or DecodeMe or 23andMe. There's a number of different companies that do this, and for a small fee, they will basically run a series of genetic tests, typically 1 million genetic tests, and look at common variability, and maybe give you some predictions about your health and possibly about predictions about diseases that you may acquire during your lifetime. Now, for the most part, these genetic tests, these companies are basically looking at common variants. And as I said, these are susceptibility loci, and the variants that they are examined don't necessarily segregate with disease down the family line. For the most part, they don't. And a recent study I should point out, this was done by Neg et al. And published in Nature in 2009, basically took the same individuals and sent these individuals' DNA samples to a variety of different companies, including 23andMe and Navigenics and a couple of others, and basically looked at their predictions based on this genetic testing around disease risk, and showed that for the most part, the tests either disagreed, as you can see here, in terms of, for instance, this individual female A, her risk for psoriasis predicted by one company said it was less than the relative risk of the population, and the other company said it was more. Sometimes the companies agreed, sometimes, but 50% of the time they disagreed in their risk estimates. So this is a problem with the specific variants that are tested, which, like I say, aren't that deleterious, aren't that pathogenic, don't segregate down the family line, but nevertheless, in very, very large studies involving tens of thousands of people, can be informative in aggregate when you're looking at the frequency differences across thousands of people. I'm going to focus most of my work on mutations that segregate down the family line, because I think they can be much more informative about what the fundamental causes of Parkinson's disease are, and much more useful, if you like, as targets for neuroscience and for the pharmaceutical industry to develop new drugs. Any neuroscience we do, we want to build upon the most solid foundation we can, and I think genetics provides that foundation. So genetics became a focus of Parkinson's disease really around 1997, 98. So it's only really been going for the last decade or so. So it's fairly new discoveries. I'm going to tell you a little bit about some of the genes that were discovered and illustrate those with synuclein and LRRK2, and illustrate why it matters as well. The field has really, really changed over the last few years, and you'll see why. So the first gene, as I mentioned, was alpha-synuclein. It's discovered in a family from Conterzi, Italy, initially. It was this particular change here, A53T, found in lots of Greek families too, and they were all shown to have this particular mutation, and they all developed late-onset Lewy body Parkinson's disease. So Lewy bodies are the pathologic hallmark of Parkinson's disease, the inclusions that are found in cells in the brain when a person dies. Since then, a number of other point mutations have been found, A30P initially in a German family, and then E46K in families from Spain. And most recently, we've actually found another mutation, H50Q, in British Columbia in the center where I work, and I think, I understand, the same mutation has been identified in Queen Square in London in a family there too. So there are still more mutations to be identified in alpha-synuclein. All of the mutations tend to line up, I don't want to go too much into the biology, but along one side of this helix of the alpha-synuclein protein. And that informs us a little bit about its function and what these mutations were doing. However, some of the big excitement came because very quickly after the alpha-synuclein gene was identified, antibodies to the protein, so reagents that will stain the protein, were created and were used in some post-mortem studies, some autopsy-based studies, to look at individuals who had Parkinson's disease during life. And what was observed was all of this brown pigmented staining. You can see it here. So here is a typical picture of a Lewy body. This is a Lewy body, a protein inclusion in the nigra, in the substantia nigra of a person with Parkinson's disease. And this inclusion is this brown staining, actually all of this brown staining that you see on the slide here, is abnormal and should not be present. But it showed us very quickly that these genetic findings in a single family initially from Conterzi, Italy, can be generalized to garden variety typical Parkinson's disease around the globe, because basically this staining, alpha-synuclein staining, has become the pathologic hallmark of Parkinson's disease the world over. It's led to a better understanding of where Parkinson's disease may begin, and Heiko Brack has a very nice hypothesis suggesting that it may start either in the gut, in the myenteric plexus in the gut, where you first see Lewy pathology based on synuclein staining, or it might start in the olfactory bulb, and then ascend basically into the brainstem, midbrain, and subsequently perhaps into limbic and cortical regions of the brain. it tells us a little bit about Parkinson's disease progression. The big discovery came, I think, in part for synuclein with the identification of synuclein multiplication mutations that were published back in 2003 to start with. A series of them have come through since then. And these show that a simple 50% increase of dose of alpha-synuclein or 100% increase of dose of alpha-synuclein, depending on whether you've inherited one extra copy or two extra copies, is sufficient to cause Parkinson's disease down the family line. And that increase in dose, as you can see here from this little part of the slide, basically is associated not just with the age of onset, but also the comorbidities or the other features that may be associated with the movement disorder. So a 50% increase in dose typically manifests with Parkinson's disease around 55 years of age. So it should be this figure I'm looking to, symptomatic synuclein duplication carrier. Whereas 4 copies, or what we call a synuclein triplication, I know it's confusing, but 4 copies of alpha-synuclein will lead to an even earlier onset of Parkinson's disease. So basically, these genetic findings taught us the mechanism that increases in alpha-synuclein can contribute directly to the pathogenesis of Parkinson's disease, and led us to start thinking about ways in which we could lower alpha-synuclein expression. And several companies now have been formed or are working on this particular agenda, many of the large pharmaceutical companies, to developing methods in which they can lower synuclein expression. Neuroscience as well has proceeded to try and understand what synuclein, the protein itself, is actually doing, because it's kind of like that alternator in the car. You've got to try and figure out what that component of the car engine is really doing. If you're going to try and understand how it connects to other components in the car engine or other genes that might be identified, for instance, in families with mutations with Parkinson's disease. So we now know that, for instance, alpha-synuclein is involved in maintaining vesicles. So these are very small membranous structures that are found in those presynaptic terminals I mentioned earlier. They typically package neurotransmitters, such as dopamine, in dopaminergic neurons, and allow those neurotransmitters to be released when these little vesicles fuse against the plasma membrane and release that neurotransmitter into the space between neurons to allow signaling, communication from one cell to another. The issue with alpha-synuclein is that it helps stabilize these vesicular structures, and we believe it's got a role not just in maintaining the what's known as the readily releasable pool of synaptic vesicles in dopaminergic neurons, but we think it may also have a role in the reverse phenomenon, which is basically how these membranous structures then basically form back into vesicles again after they've released their neurotransmitter into the synaptic space, how they basically form once more and then are available again to package new neurotransmitter. Again, I don't want to go too much into the details. I think it might be a bit too much, but this membrane basically in neurons is critical. It's polarized. It carries an electric current. It has thousands, if not tens of thousands, of very specific receptors. It has to be exquisitely maintained, and its constituents have to be maintained. And this process of basically membrane formation and membrane recycling, around vesicular recycling, I think is one of the central tenets of Parkinson's disease that is becoming more and more of a focus in terms of what the genes are telling us. Moving on to the next major gene for Parkinson's disease is leucine-rich repeat kinase 2, LRRK2. And this was a staggering advance in many ways because, really for the most part, because of its frequency. So many people in the world have LRRK2 mutations or have common variability, genetic variability. So LRRK2 is also a susceptibility locus, not just a linked to disease, but also associated with the disease. So many people with Parkinson's disease have variability in LRRK2 that contributes to their condition. So therefore, studies on it are very important in terms of if we can develop therapies for LRRK2 patients. I think they're probably going to be generalizable to people with garden variety Parkinson's disease. The first family identified that I was working on with was from western Nebraska. You can see here, Parkinson's disease segregates down the family line. These are kind of the forefathers of the pedigree here. Individuals in black have Parkinson's disease. This is that person's spouse. They went on to have several daughters, daughter here, daughter there, several, actually, I think it's 5 affected daughters and 1 unaffected daughter with Parkinson's disease. And you can see how Parkinson's disease segregates down the family line. Several autopsies conducted from the generous gift of family members when they died, and showed that the family had both Lewy body Parkinson's disease as well as tau Parkinsonism. So again, I shouldn't go into too much due to time forbids, but I'll move on. One of the things that we quickly discovered, this mutation, first of all, was an R1441C mutation in leucine-rich repeat kinase 2. We quickly discovered many other mutations, and probably the most exciting to the field has been this particular variant G2019S. We first found this in families from the coast of northern Norway, series now around 13 kindreds, and realized quite quickly, of course, that they're all related. But in the first paper, we highlighted there are other families in the United States with this mutation. Perhaps they were Norwegian immigrants. We also found individuals in Ireland, from Ireland, and also Poland with the same mutation, and not just the same mutation, but the same piece of DNA around that mutation. So these individuals, whether they were skiing in Norway or eating, sauerkraut in Poland, occurred to us they all are descended from a common ancestor, a common forefather. Inheriting a G2019S mutation increases the risk of Parkinson's disease by around 10-fold, and it is very common in some parts of the world. The autopsy series that were done showed that the majority of people with G2019S LRRK2 Parkinsonism had typical Lewy body Parkinson's disease. As you can see here, both in the midbrain and the substantia nigra, but also in the olfactory bulb and also in the gut lining. They also had synuclein pathology, typical for Parkinson's disease. We quickly learned in 2006 with these papers coming out that the spread of G2019S was actually much further afield than just Norway down through sort of northern Europe to Poland, if you like. It was shown that in Ashkenazi Jews, 14% of patients of Ashkenazi origin have a LRRK2 G2019S mutation. And across the coast of North Africa, all the way from Morocco over here to Algeria, Tunisia, Libya here, that Arab Berbers in these populations, typically 1 in 3 patients that comes into the clinic, for instance in Tunisia, has a LRRK2 G2019S mutation, which is a major risk factor for them developing the disease. We ascribe this, and it's just a hypothesis, but to the Phoenician Empire and the trading of this seafaring nation across the Med. And there's a picture here of Queen Dido, if you're interested, and Carthage, which is one of the major trading settlements, which helped to spread, if you like, this mutation. And the Vikings perhaps had some role in this, but I stick to genetics, I think. In terms of risk estimates, we've estimated this in Tunisia in the Arab Berber population background, and we find here that principally if you're a carrier of the disease, by the time you reach 70 years of age and you're a gene carrier, your risk of becoming affected is around 80%. It's a very high risk in Berber Arabs from Tunisia. However, other studies have been done in America and Europe and find risk estimates that are much, much lower. And there's a nice little review of this by Stefano Goldworm in Movement Disorders in 2011. And the risk estimates are quite variable depending on the ethnic background and the study design that was used. Many of these studies, however, are very small, very few numbers of patients, underpowered, and I think conclusions, the confidence intervals are quite wide. We've been doing some more studies in Tunisia, a much larger population series, because of the prevalence of G2019S. It is such a major health burden in that part of the world. And And we find that we can reproduce our risk estimates. So again, here you can see age on this axis. You can see the risk of developing the disease. Around 70 years of age, your risk of getting the disease is up here in the 80% kind of people at 70 years of age. The median age of onset for Parkinson's disease, if you're a LRRK2 carrier, will be affected. We're doing studies in families with the help of many, I think more than 200 families in Tunisia are engaged in this, and all their family members. We find similar risk estimates within the family-based studies. In terms of common variability, this is a study we did last year in 2011, engaging 15,000 subjects took part in this study, and we looked at all of the common variability in LRRK2. So not necessarily pathogenic mutations, they were excluded, but common variability, common changes in nucleotide changes within the gene. And sure enough showed that there are many risk factors, single point variants that are frequently found in many different populations that contribute either to disease risk or to brain health, making this an incredibly exciting target for the pharmaceutical industry because the treatment they develop is likely to be generalizable. Mouse models have been made, and there are many now mouse models of Parkinson's disease. So one thing is finding the gene, the next, and the mutation. The next is trying to understand what it does, how it does it, and making a model that will inform the development of a drug to put the condition right. This is one of the mouse models we made around LRRK2. Actually, no, it wasn't our group. Actually, I misspeak. It was C.J. Lee's group, and developed an R1441G LRRK2 mouse model. This is a mutation that's particularly prevalent in the Basque community of northern Spain, and the mice develop a motor phenotype, a movement disorder, and they also develop, in this case, tau pathology, and also have problems in dopamine release. So they recapitulate many aspects of the disease. Since then, many drug companies have started to get involved, in academia, of course, too, is driving this, and the Fox Foundation, I should give them some credit for this. They've also been pushing this agenda very hard to developing inhibitors of LRRK2 kinase, with the view that if we can the idea is LRRK2 kinase is overactive, and if you can inhibit the kinase portion of the molecule, you may actually be able to slow down the progression of the disease. I'm going to move on to most recent genetic insights because I've probably been talking too long, but I wanted to highlight a common theme, joining the dots between these genetic players. So I talked about synuclein and LRRK, but I haven't talked about the 3 or 4 latest genes in Parkinson's disease. And this is the first one I'll mention. It's known as VPS35. It's a protein that's involved in synaptic vesicle membrane recycling, and we're particularly interested in its role in what's known as the retromer. So this is quite complicated, but it's basically a pathway within neurons that helps to recycle membrane and membrane-associated proteins within the neuron. We're focused in those presynaptic terminals, but you can look. A lot of the biology of this has actually been done in the cell body and in non-neuronal cells. And this is the first gene. It came out last year and has been reproduced. So there are quite a lot of families now with VPS35 mutations. We've recently described another mutation in the gene known as DNAJC13, also known as REMI8, receptor-mediated endocytosis 8, in families of Mennonite descent, again with late-onset typical Lewy body Parkinson's disease. The picture here is of Rembrandt, who may or may not have been, but is alluded to have been a Mennonite, one of the earliest Mennonites. And then there's a paper, a fairly recent paper, of a very small family that came out in PLOS One just fairly recently, of another DNAJC mutation in a different DNAJC, DNAJC6, again involved in this process of synaptic vesicle or membrane protein, membrane vesicle membrane protein recycling. So we're starting to get what I think is a common picture, and here's an illustration of it, of what's going on in Parkinson's disease in terms of its cellular pathogenesis. And some folks have been focused up here in terms of synuclein, for instance, and stabilization of snares, and perhaps synuclein's role in maintaining synaptic vesicles in this area. Others are interested in these tubular-like structures, the VPS35 and the retromer, a lot of work is focused up here on lysosome and the breakdown of alpha-synuclein aggregates that you find in Lewy bodies in Parkinson's patients. But basically, principally, LRRK2, I should point out, is found here, and it's involved in the sorting of proteins in the trans-Golgi network to different parts of neurons. But principally, all of the proteins, all of the genes that encode proteins in Parkinson's disease fall along this common pathway. And I think it's becoming clearer and clearer what the picture is in terms of how to join the dots, how this motor actually functions, and what are the connecting routes between the major components of it, each of which becomes a drug target. So the last thing I'm going to illustrate is how advances in genome sequencing will speed up the development of new treatments, and how you, or people with Parkinson's disease, or people interested in helping people with Parkinson's disease can help. And that's really to point out there's been a real revolution in genome sequencing, tremendous revolution in the last 3 years. It's now possible, and we're doing this in our lab, to sequence hundreds, if not thousands, of genes in a single experiment, and to look at all of the variability and how it contributes, for instance, to human health or to human disease. This illustrates a panel, for instance, that we've made that captures all of the genes in which mutations have been found that are linked to neurodegeneration in general, not just Parkinson's disease, but dementia, motor neuron disease, for instance, and peripheral neuropathies. They can be assayed in a single experiment and we're doing this for something in the order of about $150 per subject, very, very cheaply to identify risk variants for disease. And working with these individuals and their family members, we're starting to get a really clear picture of genetic and clinical correlations, and also correlations with treatment and treatment outcomes in those individuals. And this is something that's going to be a long-term effort, a longitudinal effort. It's going to involve many, many people and many, many families, and the generous gift of their time and data and DNA samples, blood samples. But ultimately will, I think, give us like I said earlier, the solid foundation that we need for neuroscience advances and for therapeutic advances to really stop this disease's progression, not just to create drugs that are good for the symptomatic effects of Parkinson's disease, but drugs that actually slow its progression or stop its progression in its tracks, and in effect provide a cure for the disease. So I think leave that with some questions and answers.
Okay. Thank you very, very much, Dr. Farah. That was really superb, and we all appreciate it. Speaking on behalf of our entire audience today, I know that they will agree with me. This is a wonderful talk, much appreciated. We already have quite a number of good questions in. You're invited to send others. If we don't get to them by the end, we will certainly try to get back to you later. I have taken the editorial liberty of not including all of them because some of them were submitted before Dr. Farah's talk, and I think they were well answered during it. So if you don't hear yours, don't think I'm ignoring it, but I think it was probably well covered. There's a group of questions, Dr. Farah, to kick this part off that I think clearly show there's a tremendous interest among our people in our audience today in how they can go about participating in research, which, of course, is music to your ears, and you've said that in several different ways. One kind of question asked by 3 people is the commercial services. You talked about 23andMe. You talked about another couple of them. Another question from another person was, what about medical centers? Instead of going to a commercial service that will give you a personalized report on your genetic risk, you would then go to a medical center, which, like the University of British Columbia, which has a sterling genetics program, and give your DNA there and contribute to their research in that way. Do you have any additional comments, additional to those you already made, on what one would do if one would like to participate in research, would like to contribute DNA to this whole thing? What sorts of places would you, if you were in that situation, go to first?
Well, yeah, very good question. So I'm really interested in getting neurologists as part of the equation. One of the, I think, problems or challenges with direct-to-consumer companies and that type of genetic approach is that it kind of marginalizes the role of the neurologists in both the diagnosis of the disease and the follow-up exams, and of course in terms of the treatment. And so I'm really keen to get individuals to take part through academic medical centers. There are a number of big groups doing genetic work in Parkinson's disease, and a quick Google search on Parkinson's disease genetics, and we'll probably give you a handful of these. They're scattered around the world. I'm part of a consortium known as the GEOPD, G-E-O-P-D, which basically is working right now as something in the order of, I forget the number, but it's kind of like 20 or 30 different countries around the world, something in the order of 60 or 70 different major neurology centers in those countries, and currently engaging more than 60,000, maybe 70,000 individuals in the studies. From my own personal point of view, we don't have all the money in the world, and we don't have all the time, but my center is incredibly interested in individuals who may have atypical Parkinson's disease, atypical forms. We're very interested in individuals who have a family history. I should say 1 in 7 people with Parkinson's disease have a family history of the condition, typically in a first-degree relative. We're very interested in hearing from you. And to get engaged in the studies, we have a worldwide consortium. We may not be able to see you directly, but we certainly have a friend, a colleague, probably in a country and a town near you, who we could recommend and suggest that you hook up with to get engaged in our study or related studies in those centers. So it's pretty easy actually to get involved.
Great, thank you so much. This second question is really a subset of the first, but it will be a quicker one, I think, for you to answer. That is, a gentleman is calling and saying he's heard that there's an age limit on the usefulness of participation in trials like this. He's heard, and I don't know where he lives, this particular person didn't identify that, but he's 80 years old, and he's wondering, is there any age limit that would limit the people who could actually participate in a research study, genetic research study?
Not in a genetic research study, no. Perhaps from a clinical trials point of view, in a medication study, yes. And often medication clinical trials are looking for de novo or new patients who are very recently diagnosed and perhaps have not been on therapies. So one of the problems for a lot of research studies is that taking medication for Parkinson's disease can cloud some of the answers to the questions that our investigators are looking at. But medications don't affect your DNA, not the DNA that you inherit. And so no, people of all ages can participate in genetic studies. One caveat is that it becomes increasingly more difficult to diagnose Parkinson's disease in the very, very elderly. So that's an 80-year-old, I think, is a fairly young man. But in the very elderly, it becomes more difficult to diagnose. People are less active and less mobile generally, and it becomes more difficult to make the call on the diagnosis. But that's the only caveat.
Thank you. Every good group has its ringers, and I strongly suspect that I know the identity of the person who asked this question. I may even out him. I think it is Dr. Heydrich from Washington, D.C., and if I'm wrong, he will scold me afterwards. But his question, he's an MD, by the way, so that's why he's a ringer. He asks, can gene expression be altered by nutritional practice?
Well, almost certainly, I think. People often ask me, what would you do? I mean, it's all very nice studying these genes and finding these mutations and setting up these models and pushing the pharma industry to create therapies. Well, what can I do immediately in my lifetime to have some benefit? And the answer to that, really, I think you've had a show on it previously, is exercise. I think exercise is one of the best neuroprotective agents out there, if there is one. I would say it's exercise, and get engaged in some sort of exercise program, whether it's swimming or dance or something like that, I think is incredibly beneficial. So yes, and I think exercise probably does, and diet and nutrition does affect gene expression. One of the central issues in the genetics is pointing us to is this, what's known as endocytosis. One end of endocytosis is a process known as autophagy, how cells degrade proteins and basically help them to be turned over, so new proteins are synthesized, old proteins are degraded, and the degradation machinery is known as the lysosome. This breakdown of proteins, as well as the creation, the translation of new proteins, is regulated by something known as the mTOR pathway, mammalian target rapamycin pathway, and that is influenced very strongly by nutrition and by exercise. So I think, yes, that was a very good question. I think gene expression can be altered by diet, by nutrition, by exercise, with beneficial for Parkinson's patients.
Good, thank you. Here's a question. I don't think you answered this. If you did, you'll forgive me. Gentlemen would like to know what of all people with Parkinson's, what percentage of them have a genetic history of the kind that you were listing, one or more of the genes that you were listing that are possibly inculpated in Parkinson's, compared with those who have no such genetic history? What's the ratio there?
That's a really good question, and it's increasing by the day. So it's a difficult to put a figure on, but in terms of family history, so that's not necessarily genetic, but because families can be influenced by a common lifestyle, dieting, for example. But in terms of family history of Parkinson's disease, it's 1 in 7 individuals have a family history of Parkinson's disease, 14% or so, with the fairly recent Mayo Clinic study. In terms of the number of those individuals that have a really serious, deleterious, pathogenic mutation that segregates down the family line, it's only a handful. I would say something in the order of 10%, something of those 1 in 7, all right, at most, so far. In terms of common variability, so the susceptibility loci, which also sometimes happen to be the same as the pathogenic genes, pathogenic mutation genes like synuclein, for instance, tau, LRRK2, many people with Parkinson's disease have those susceptibility loci. They make a small contribution to disease risk, but nevertheless are important players in terms of the disease that individual has. Many of the genetic forms of Parkinson's disease, I should have pointed out, appear to be population-specific. So when we often asked about ethnicity, ethnic background, when we ask when people get engaged in the studies that we're doing. And if people are from, for instance, northern Spain, we'll look for R1441G and LRRK2 first of all, because it's something in the order of 10% of patients with Parkinson's disease in northern Spain from the Basque region have an R1441G mutation. As I said, Berber Arabs, so if they're Berber Arab origin from North Africa, 1 in 3 patients has a G2019S mutation. And that's not 1 in 3 patients with a family history, that's 1 in 3 patients in general has a LRRK2 mutation, a G2019S mutation specifically. In Asia, the risk factors like G2385R and LRRK2 found in 8% of all Asians. And when I say Asia, I mean Japan, Korea, China, Singapore, across Asia, Taiwan. And but those risk factors like G2385R perhaps only double the risk. They don't increase the risk tenfold, but just double the risk. They're just susceptibility factors. So it's population-specific. The frequencies are increasing as our knowledge increases. And so to define this and put a hard figure on it is very difficult. It's a moving target, but it's an increasing one.
Thank you. This is putting you on the spot a bit, and I realize it's an entirely unfair question, but it's the kind of question that we get at PDF a lot, which is if I have one primary relative, mother, father, sister, brother, with Parkinson's, what does that do to my risk? And it's a general answer we've had, which I hope is more or less right, that it may double your risk if there's a single primary relative, but it doubles it from a very small level, as you were pointing out earlier. This particular person, she is asking a very specific thing, which I realize has no specific answer, but I'd love to hear you take a whirl at it, Dr. Farah. Here's the situation, probably a family situation of her own. 2 siblings, both have Parkinson's. One of the siblings has 2 children, and they have 5 children together. So one of the 2 siblings who has Parkinson's has 5 grandchildren. What generally, very generally, would be the risk, the increased risk for those grandchildren?
Well, I would say, to be honest, it's marginal. Until you actually pinpoint the genetic factor driving that, perhaps, that familial aggregation, the reason these 2 siblings have the disease in the first place, I think it'd be very difficult to put a risk estimate on it. And even when you do find a specific mutation, for instance, perhaps it's LRRK2G2019S, calculating what the risk would be in the offspring is also going to be incredibly challenging, but it's relatively low. The biggest risk for Parkinson's disease, is, which I should have pointed out at the beginning, is age. It's not genetics at all. Genetics really is the route into understanding this engine, if you like, this motor in the machine itself, and to what is the underlying substrate, if you like, that is resulting in Parkinson's disease, and what's going wrong in the cells. We're not doing this genetic study really to inform individuals about personalized risk, and because I don't think we can. I don't think we have enough information to do that. And for the most part, Parkinson's disease is considered what we call a multifactorial condition. You still have to get, to 60 or 70 years of age before you develop the disease, and there's a lot of things on the way that can influence whether you will or whether you won't. So it's very hard to put risk estimates on it. So I'm going to fudge my way around the question and not answer it.
Probably very responsibly. And
I think the point I want to make is that please get engaged in genetic research, but do it for the right reasons. I don't think we like I say, we want to understand what is the cellular pathogenesis of this disorder, so we can fix it. This is not about defining individual risk. We're looking for people who are altruistic and philanthropic, and who can make this humanitarian gift, really to society, to the world. Getting something back like a hard and fast estimate on exactly what my risk is going to be is, as I pointed out with the 23 and Navigenics study that was published, or results that were published in Nature, this is very difficult to predict, and 50% of the time the predictions are wrong.
That's such a good point, and I'd like to generalize it, if I may, beyond the field that we're discussing today, the idea of participation in research being a philanthropic act. So many of us think of it as something that can actually give us some direct benefit, and it'd be wonderful if it does, and it sometimes does. But given the number of trials in so many areas of Parkinson's that don't work out, the idea that going into the trial is somehow you've then failed because you haven't learned for yourself or you haven't got a new treatment for yourself that sets you up for something that is often unrealistic. And at the bottom, it really is a generous act going into these trials, and the science, including the science that doesn't work, also teaches people like Dr. Farah something, because failures teach as well as successes. So thank you for making that point about the generosity. I think it's awfully important. I've combined three questions here, Dr. Farah, into one, which I find very tantalizing, and it has to do with the different ways in which Parkinson's develops and the different ages at which it develops. And the questions, which I'm kind of making generic here, are, is this because of genetic subtypes? Is it that different ways in which Parkinson's manifests and different ages at which it is diagnosed, could these be traced in general, in part or in whole, to genetic differences?
Absolutely, yes. Absolutely, yes. I think so. We have good evidence for that for some genetic subtypes. So, for instance, be too specific about it, but for instance, individuals with PINK1 homozygous mutations or compound heterozygous mutations tend to have very early onset Parkinsonism. It's very slowly progressive, but it tends to be the akinetic, rigid form of Parkinson's, subtype of Parkinson's disease, rather than tremor dominant, which many are more familiar with. And again, we understand the primary risk factor, which is loss of PINK1 expression. We don't understand how that contributes necessarily to that manifestation, but definitely the genetics does underlie subtypes. Other forms, for instance, synuclein variability has a major role to play in cognitive aspects of Parkinson's disease, and perhaps more accelerated cognitive decline in patients with Parkinson's disease. So this is, again, a subtype of Parkinson's disease with dementia, which is promoted by synuclein variability. So yes, there's the answer to that. And this is something that longitudinal effort, long-term effort of engagement of patients, their neurologists, their geneticists, their a local medical center, a collaborating center, as part of this major international effort, is going to address and answer, because we can, taking this data over the course of an individual's disease, looking at the genetic profile, which doesn't change in your lifetime. You only have to have this sequencing done kind of once. But looking how that correlates with clinical progression and with outcomes, that's the data we're going to put together over the next 20 years or so. And I think we're going to have a much more rigorous and illuminating answer to that question in 20 years. Hopefully I'll be around to tell you.
Great, thank you. Here's a question that I know that you're not a genetics counselor, yet you seem to know about everything, so I'm going to ask it anyway. Here's a woman who is a sad story here. She enrolled in good faith in a genetic testing program. She couldn't understand the results, couldn't find a counselor to help her, and the people in charge didn't seem to be telling her very much unless she pushed them, and it turns out that, she didn't get very much even after that. So do you have advice for people going into genetics studies who find this kind of ready smile at the front end, but somehow a missing reporter at the other end?
Right. No, that is such a familiar story. I get it all the time from neurology colleagues who basically present, a patient comes into their office with their 23andMe results typically and says, "What does this mean?" And unveils this report of all these risk factors, and of course the neurologist is dumbfounded by it for the most part, and the patient really is none the wiser. And to be honest, I don't really think 23andMe are much the wiser either, even though they provide the prediction in the first place. It's going to take many years of effort. So I think we're kind of like, I liken it to, we're like page one of Genesis when it comes to our understanding of how genetics influences human disease or human health or human traits. And for the most part, we're very good at studying single gene disorders, and we can put fairly good risk estimates on that. But typically what you get when you go to these direct-to-consumer companies is they're not testing for specific deleterious mutations in known genes, for instance, that cause Parkinson's disease, with maybe one or two exceptions. They're testing common variability that we all have and looking for frequency differences. So in the general population, maybe this variant is seen at 10%, and maybe you have it, and maybe it's very marginally associated with an increased risk of a particular condition. Giving you a genetic counseling on that type of thing is actually meaningless. It doesn't. It cannot be done. So I think the general advice would be not to be cautious. Get engaged in genetic testing. Be very cautious if you're parting it with money to do it. If someone's trying to saying they're going to predict this stuff for you, and they're asking you to write a check for a certain amount of money to do this, I'd be very cautious with that, because I think for the most part we don't know. We are quite naive, and this is coming from an expert in genetics in Parkinson's disease in the field. So I would be very cautious with promises made at the forefront, and then the lack of delivery at the back end. There are some very good genetic counseling services. So if you do, for instance, have a LRRK2 G2019S mutation, you should be able to get some reliable advice on that. One good place to look is Gene Tests, a website online. It's one word, I think, if you write Gene Tests. It's an NIH-sponsored website that's run through Seattle. And you can look up specific gene reviews, which will tell you about that particular gene, the mutations in it, and their contribution towards disease risks, as well as companies that do that type of testing on a fee-for-service basis. It's not genetic counseling one-on-one, but it's the best that we can do through the web kind of thing or remotely. So have a look at that.
Great. Thank you very much, Dr. Farah. This last comment actually prompts something, if you'll forgive a brief public service announcement for Parkinson's Disease Foundation. We have a program, I know many of you on this seminar are familiar with it, called Parkinson's Advocates and Research, which is now a 4-year-old attempt, a very successful attempt, I must say, to create a leadership group of lay advocates in Parkinson's research. And one of the things they do, because we've come across this and it's prompted our concern in the past, is work with people who are running trials to make sure that the people who participate in the trials, including genetics trials, are given the best possible respect and treated actually as partners in the enterprise. And this lady's comment, very thoughtfully responded to by Dr. Farah, is just one of those cases in point. And the research process doesn't seem to be quite regarding its main champions and participants, namely the people, with the kind of respect that they deserve, but also that will help advance the science and get more people involved. We have now 200 people in this ambassador group working around the country in these areas, and it just reminds me how important this work is. Did you have a comment on that, Dr. Farah?
Yeah, well, a few comments. Probably I've got comments on everything, but I agree with you wholeheartedly. It's very difficult, though, on the other hand, to write to each individual and tell them, about advances. So what I tend to do, and our group does, we work with our local Parkinson's disease support group here in British Columbia, and they have a newsletter that they send out every 4 months, and basically we provide material for the newsletter to say what's recent, how individuals have contributed, and how that's driven the science forward. And we write about very specific genetic discoveries. If we find something deleterious in a person's genome related to the disease, we do contact them and ask them if they want to know, with appropriate ethical consent from our research ethics board. But I agree with you, getting individual participants and researchers together and engaging people in this, and neurologists, engaging them in this long-term basically dialogue, this interaction, which is what we have to do if we're going to move this forward, is critical. And one of the things I loved, and I know that you've got a big part to play in this, is the World Congresses of Parkinson's disease, where you've got this patient and neurologist and neuroscience kind of forum together. And the meeting in Glasgow was marvelous, and the meeting in Montreal, I'm sure, will be just as good. So I'm really excited about that. You have a patient advocacy forum. It would be good to, to hear from you or the patient advocacy forum about what people like me, researchers like me, could do to put the word out and to fix problems in terms of communication and feedback where you see that there is a problem. We'd be very interested to hear more.
Great. Thank you very much. We're running over. I hope you'll forgive us. This is a very rich subject, and I will take the editorial privilege of giving it just 4 or 5 more minutes. First of all, a comment, and then one more question of Dr. Farah. A couple of people just wrote in, Dr. Farah, and I will take the liberty of answering this, and you'll tell me if I'm wrong, saying they thought they heard you say that you don't get Parkinson's until age 60 or 70. I believe the answer to that is, to whoever wrote this, actually the 2 or 3 of you, that Dr. Farah was referring to the average age or the median age, which is indeed the late 60s or early 70s, and that, of course, there are many people who are diagnosed later than that, and many, many people who are diagnosed much earlier. I think there are 10% diagnosed before the age of 50. So he was just referring, I think, to the average or the median. Is that correct?
The median age is a statistical term, but yeah, you're absolutely right. It's a very broad range. Our youngest patient is 7 years of age, to give you an example, and our oldest is probably in their mid-80s.
Yes. Thank you. Last question. I love this one. Why did you go into this field?
Yeah, that's a good question. Well, I've been in it for a while, but I'm very interested in brain and brain function, and I'm very interested, obviously, in genetics. And I thought to myself, there is no better approach, I think, in looking at the human brain than looking at genetics, because you can get access to the information you need, the root causes of the problem, basically without taking a biopsy. People with brain disorders are already suffering enough in terms of their cognitive ability or movement ability or whatever, and you can't take biopsies of living people of brain tissue. So I thought this is a wonderful combination. I got engaged in this very early by actually working in psychiatric hospitals when I was a child, and seeing how disabling the medications were to those patients who were in long-term care in psychiatric hospitals, and how little efficacy they had, and how basically how disabling they were to those individuals. And I thought there's got to be a better way than this. Most medications are not developed around the cause of the problem. They're developed around the symptoms. And so I've been basically applying my skills of that since I was a teenager and going into this route.
That's a wonderful way to end this, and thank you so much for the privilege. You've really done us proud, done us all proud, and we're all very, very grateful.
It's the other way around. I'm grateful to have the opportunity.
I'm glad you enjoyed it. A couple of more announcements, if I may. First of all, there is a wonderful article in the very extremely well-known Parkinson Disease Quarterly newsletter called the Parkinson Disease Foundation News and Review. It is in the winter 2012 issue. That's 2 issues ago, and the author is one Dr. Matthew Farah. So if you want more from the same source and didn't exhaust your enthusiasm with this terrific talk, if you haven't got if you're on our mailing list and don't have the newsletter, email us or call us, and of course we'll send you the article. And I think you'll find it as enriching and informative as I did. A few other public service announcements. If you haven't already done so, please look at the online survey. It should be on your screen, I'm informed. We really, really, really use the feedback on these webinars to ensure that we plan well in the future. We don't just pick randomly. We go on the basis of the need, the expressed need, how long ago we did something, how well we did it, and how we could then improve it. And all of those questions go into this, and your feedback is terribly important in that regard. So please, before you leave, I think it takes you about a minute and a half probably to do it, but please do it. We'd really appreciate it. A thank you again to our terrific corporate sponsors for this series that we're now beginning with the genetics story. Abbott Laboratories and Teva Neuroscience, 2 model companies in the promotion of public awareness in our field, and we're extremely glad to work with them and welcome their financial support that really does make this whole thing possible. For those of you who would like, or those of you who have friends who would like, there's an archive of the event. I think you're probably familiar with this at this point, and it will be available 1 week from now on Tuesday, October 2nd. Just go to www.pdf.org. We will send you an email when it's available so that you can listen to it again, let your friends know about it. Please note in your calendars, your diaries, the second in our series, in our current series, which will be on Tuesday, November 20th, at the same time, 1 p.m. Eastern U.S. time. And the subject will be caregivers and Parkinson's, and the speaker will be Julie Carter, one of the most experienced RNs in the field and professionals generally in our field, from the University of Oregon, and she will be leading that. So we'll all look forward to that one. So I want to thank Dr. Farah for this wonderful talk and reducing one of the most difficult subjects to manageable bites and edible and digestible bites. We're very, very grateful for this, and we are, of course, very grateful to all of you who've joined us for this whole show and are, I'm sure, the richer for this experience. Thank you again, Dr. Farah. Thank you, everybody, and enjoy your autumn season, and we'll be, I hope, speaking to you again very soon. Bye-bye.